This figure is showing us the power of PCR for amplifying or exponentially increasing the number of copies. So remember, the purpose of PCR is we start with a needle in a haystack. We're going to use PCR to make so many copies of that needle that ultimately we have a bunch of needles with a little bit of hay in it. So remember, with PCR, we start with our primers and our template DNA and DNA polymerase. We run through three phases to each cycle: denaturation by heating, primer annealing, where we let the temperature cool down to approximately 55 to 60 degrees. That allows the primer to anneal to the template DNA or bind on the basis of complementarity. And then we have extension, which is where the DNA polymerase comes in and extends the DNA to which the primer is bound. So at the end of the first cycle, we've gone from 1 copy to 2 copies. At the end of the second cycle, 2 to 4. At the end of the third, 8, 16, 32, and so on. And after 20 cycles, we have over 1 million copies of that particular strand of DNA. So that's the power of PCR. It allows you to make lots of copies of a specific region of DNA. In this case, the region we're amplifying would be an STR, so that we can separate those fragments on a gel and visually observe differences in size between them, and we can use those differences in size as a DNA fingerprint profile.